Hello everyone. I'm glad to see a lot of our gene regulation friends today. All right, so today we have a big program. Just let's start. Let me share the screen right away. My name is Alexander Kell, and I will talk today about gene regulation and specifically about generegulation.com. And let me share what I'm talking about. All right, I guess you see my screen. All right, so today we are going to talk about this generegulation.com. It's a free TRANSFAC website where you can register and do your analysis. We also will look at another free tool, which is called free platform, free GeneXplain platform, which you can register. So at this website. And we'll talk about the difference and what you can do with generegulation.com to study your promoters. All right, so but before, well, let me just start right away and do the first search, and then we'll talk about what we're actually doing. So as you know, if you type generegulation.com, you're here. And you have to register here for free, and you get your email, your account, your name and password, and then number of programs are available for you to understand gene regulation. And basically, one of the first program, which was published many years ago, which is called Match, and we'll just simply run it. So you see this very colorful interface. What we are going to do is simply take one sequence of that promoter of that gene, which is CXCL1. It's a chemokine ligand, and we'll talk about that, why we took this gene a bit later. Just by take this sequence, copy it, and put it here. And then we'll basically select vertebrates. We are talking about analysis of human gene, and we'll simply start the analysis. So let me basically make a bit smaller font so you see all the buttons. So we submit the form and we immediately get the results. So we actually got this nice table, and each line is one predicted site for transcription factor. Here we see the transcription factor names. Here we see these identifiers for the matrix, and we'll talk what it is. And if you click on graphics, you see this nice interface where you see your sequence, your promoter, and in a very simplified manner, you see where you found your binding sites for transcription factors. All right, so before going into details, let me actually talk a little bit more what we are in fact doing. So for that, I will start my presentation. Don't worry, it's a couple of slides, but you need to understand why you're doing this. And actually, I need to understand it first of place. All right, so we are talking about searching for regulatory elements. If we talk about in a very old fashion, so basically we know that in front of the gene there is a promoter area, and this promoter area very early in the history, people understand that they contain some sort of regulatory elements which are involved in regulating gene expression. Basically, they are involved in letting gene transcribed, or blocking the gene from transcription, repressing the gene transcription. And but very important to understand, we are talking about the start of transcription, which is in front of the transcribed area, which is later is spliced, and it's different from start of translation, which is here. But we are talking about start of transcription, and then in front of it, there is a promoter area. And very early picture here from some very early review where researchers collected so-called boxes like Tata box, cut box, and inner element near start of transcription, to characterize a little bit this region to understand what is common between these promoters between different genes. And later on, it was clear that there are a lot of other proteins which are involved in regulating the gene expression. We are not talking about some kind of boxes. We are talking about positions in genome which are sites for binding of specific proteins which are called transcription factors. So basically, nowadays, the idea of these gene regulatory regions of the genes are much broader. So actually those regulatory regions are not only promoters, but also enhancer regions, which can be very far from the start of transcription, or can be even in enhancer or in introns, or in the regions between the genes. And those regions, enhancers in different places, as well as promoter regions, they contain binding sites for transcription factors. And the transcription factors are proteins which are acting in nucleus, and they're binding to DNA in a specific manner. So which means the protein has a very specific, ability to find a particular sequence in a DNA and bind to it. Right, and till now, it's kind of, question and a puzzle how this binding is actually controlled. So what we are doing, in fact, we are simply collecting all cases from literature, from experiments, where we observed that a particular transcription factor, let's say E2F1, was found to be bound. And there are various experiments how to find it. So currently people are doing, these, very precise ChIPseq experiments. But early time and still now, there are much more different experiments to validate not simply binding of that factor, but also that after the binding, these transcription factors start regulating the gene. So actually push the gene to express in particular conditions. So what we are doing is actually we simply collect those cases from different experiments. And in order to understand what is common between all these sites, which would characterize perhaps the ability of that factor, of that protein, to bind and to find those sites. And very early we found out that those sites are very variable. So which means that there is not like one particular sequence which these factors are always recognized. So there is a variability in the nucleotide positions. Some nucleotides, some positions are very stable. So like, for instance, the sites for E2F1, they are characterized that some nucleotides like this T or these two CGs are very conserved, very not variable, and they always the same. But other nucleotides can vary, and some of them vary very much. So very, very much. And that's why here on this logo plot, there are the smaller letters, which means there can be C or G, or maybe sometimes you can have even more variability among 3 nucleotides or even 4 nucleotides with different weights. So, and this model of the binding, which is called positional weight matrix, which you can see here, is constructed simply by counting how many times you observed a particular nucleotide in the particular position of that site. And of course, it's very simplified version of a simplified model, although it's very robust, and till now we are using it. And why we need that? Because we need actually to predict new sites. And that's exactly what Match program is doing. That's exactly what we just did, and I will repeat it again. The program takes the library of these positional weight matrices from TRANSFAC, and then it searches for matches. It's actually scanning the sequences which you gave, and it finds those positions in the sequence where the nucleotides are actually fitting to this matrix. And this score of that match is calculated by this formula. And the very important part of the match algorithm is actually this information content. As you can see, this I is present here in that formula. It gives additional weights to those positions in the matrix which have high, conservation or high, very low variability. And those positions contribute to the score most, and the rest of them, of the positions, they contribute to the score less. And that's a difference compared to other algorithms, where these contributions in other algorithms contribution is considered equally. Here it's considered not equally. And we showed some time ago that it gives certain some advantages for some transcription factors. So there are papers about that. So basically, that's the core of the algorithm. And why do we need to find sites? Because, not far than knows, there are still not enough sites known in the genome. Although you perhaps know that there is a lot of ChIP-seq data which are actually available and coming more and more, detecting in different conditions the binding properties and binding positions of the transcription factors in various genes, in various genomes. But if we calculate and estimate how many sites are actually acting in genome, we can say that still there are more than 90% of binding sites are unknown. So you still have to predict binding sites using some some kind of models. For instance, these positional weight matrices. Or we also will look at the two other programs which are available on generegulation.com, which apply a little bit modification of those of the algorithm. The program called PMatch and AliBaba 2. So there are two kind of advanced versions of such an algorithm. But we'll come to that a bit later. But first, let's talk about this a little bit complicated thing. So before that, we'll return back to actually to the interface of match. So actually, let me return back to this interface. And you see here at the right side, so here at the left side, we put our sequence. And at the right side, there are several, cutoff selections. And the cutoff is, for instance, here it's clear saying that the matrix cutoff is 0.7 and the core cutoff is 0.75. So what it means that actually, if we look at this slide again, it's here, right? That actually the binding sites, the matrix, has the most conserved parts. Well, sometimes they are actually split, like in this case. So these three nucleotides are split by not conserved part, but often the binding sites have a core which is most conserved. And there is and then in this algorithm, in the match algorithm, we actually split the score in two parts. First part is called the score of the core. So the 5 most conserved nucleotides inside the sequence, inside the pattern, we call it also the motif, and the overall score, which is calculated for the whole length of the motif. So, and for each of that score, we have a cutoff. The cutoff means if the score of the site is higher than the given cutoff, then we claim and we detect the binding sites for that transcription factor. And of course, these cutoffs are very much arbitrary. We don't know which of them are actually right and correct in different conditions, and we'll talk about that a bit later. By the way, actually, let me tell you that In fact, the floor is open for the questions. You have here in the chat, you have possibility to put your questions, and there is a Q&A section. Yes, Q&A section where you can put your question right away. And we'll have time for answering the questions at the end of my presentation. I'm going to do it maybe around 40 minutes, and then we have enough time to answer all your questions. You can also put your, if you don't understand anything or some particular thing is not clear to you, you just can put question right away in this Q&A, and I will try to, watch on those questions and answer them right away. Okay, so coming back to this cutoffs. So basically, we made an attempt to put 3 standard cutoffs for each of the metrics in our library, and they serve for different purposes. So we will demonstrate that simply on the case of this sequence. For instance, the analysis was done with cutoffs which are called minimized false positives. And if we run it, we see that we found 12 binding sites in that sequence. If we select the minimize false negatives, then the list of the predicted sites will be much bigger, and we found actually 403 sites in that sequence. And we actually, if we look at the graphic, we see that those sites are predicted overlapping a lot, and basically each position of that sequence is covered by many different predictions. So it looks like a lot of them are maybe fails. But let's discuss that now in more details. So I'm returning back to my slides, and here we have this curve. So let's take one matrix. So for instance, we are talking about this E to F Q6 matrix. And here in the TRANSFAC, we have this matrix, and this is which matrix was built very early, in 2000 actually. And the number of binding sites which were used to build this matrix was not a lot at that time. So you see in total it's how many? 6, 8 sites in total, and then the nucleotide distribution is presented here. But okay, this matrix can be used to find new sites for E to F, and for different family members of E to F family actually. And then what we did, we actually took all known binding sites and applied this matrix to them. And we can set a cutoff. Basically, if we put a cutoff around 0.75, then the false negative rate is actually 0. So which means that we predicted all binding sites which are true. But with such a low cutoff, when we applied this matrix to completely random sequences, or in fact, we applied them to sequences from exon 2, so which are very rare, contain any binding sites for transcription factors, we can assume that all binding sites which are found there are kind of false positives, and we can say that there is a lot of false positive, and we put it as 100%. And then if you set a cutoff higher, say around 1, then we will not found nothing among the real sites. Not a single binding sites, a true site, actually have a score 1.0 in this case. So in that, it means the false negative rate is actually 100%. So we lost all sites. But then we get a very little false positive rate. So we found not many sites in a random sequence. And then if we sum up these two error rates, then we can find a position somewhere here in between where the sum of these two errors is kind of minimum. So we call it the mean sum, minimum of the sum. All right, so there is a question about one particular gene. I think we'll come to that question a bit later. All right, so good. So that's with this knowledge, now we can come back to this interface, and that's exactly what's happening. So if we select the minimized false positive rate, so you find very few sites, and those sites are actually very high scoring. You see here the score, the core match and matrix match score, they're all around 1.0 or 0.93. So they are very high, and very few false positives are actually expected. And if we apply now minimize the sum of both rates, which is kind of a optimum, average optimum, so we find an average number of sites. You see, we found actually 51 sites in this sequence. So they're sometimes they're overlapping. It's kind of hotspots, and we'll talk about this hotspot a bit later today. And some positions where those sites are found. All right. And so that's why the recommended cutoff, which are thresholds, which are recommended to use if you have no idea what you actually want to do. Just want to basically find some sites in your promoters, then the recommended is to use the minimize the sum of both error rates. But actually, the problem is not so easy. First of all, what we are going to do, we are going to actually put here a completely random sequence. All right, so I have here generated a random sequence. Right, not here. Let me find it. All right, here. It's a completely random sequence. What means random? Basically, I took a random generator which generate 4 nucleotides in random manner. The only thing it does, it actually take into account the nucleotide composition in human genome, which is not even actually. If you look in different regions, for instance, in regulated regions of human genes, the nucleotide compositions is not really even. Taking this nucleotide composition of gene regulatory regions of human genes, I generated this sequence. So, okay, so we expect basically that this sequence is not regulatory because it's simply random, right? So let's reset the form and put here this random sequence and run it with minimized false positive rates. So again, selecting vertebrate species. And okay, yeah, we found only 3 sites, but still we found some sites even in a random sequence. That's even already, cling the bell, ring the bell. But actually even worse, if we select, for instance, false negatives and submit and look at the result, do you see any difference to the picture we observed when we analyzed the real promoter of a real gene? I don't see any significant difference. Basically, practically every nucleotide in that random sequence is covered by at least one prediction. And that's really a problem. And in fact, it's the truth. Basically, the promoter regions in human genome are not very much different from a random sequence. And that's for purpose. In fact, because they have to work in a very, very many different conditions. But we'll talk about this a bit later. So there's another question we have. Okay, that's again about that particular gene. Okay, we'll talk about it a bit later today. All right, so then there is a problem how to discriminate the real promoters from random sequences, and also how basically find those transcription factor binding sites. It makes the promoter working in particular conditions, for instance, in particular tissue. But before coming to that, let's look at the other other algorithms which we have here. And maybe they are better. So they may be having less false positive, for instance. And let's consider one algorithm which is called pmatch, which was published in 2005. I will show one slide about this pmatch algorithm. So pmatch, it was published in 2005, and the idea of that algorithm is the following. It's presented in this picture. Basically, if we have true sites which we collected from different experiments, and each dot here is represent this true sites in some imaginary space, like multi-dimensional space. Then a positional weight matrix can be considered like a sphere in that space with some center. And basically the center of that matrix would be the perfect consensus. And as you can see here, it can happen, and it's often the case, then none of these real true sites actually fit to the perfect consensus. But that's only one problem. But the second problem is that it's a sphere. Well, it's kind of sphere. But the true distribution of these true binding sites in that imaginary multidimensional space is maybe not even. So which means basically using this matrix, we would predict some sites which are in that area, for instance, although there were none of true binding sites were actually found in that area of sequence space, right? So which means maybe these sequences which we found here, those sites which we found in this sequence space are not true sites. They have some kind of property in their sequence which is not well described by the simple model of positional weight matrix. And that's why we predicted those as true sites, but they are not really true. So the factor is not binding to them, or maybe it's binding but not regulating the gene. So then pmatch idea is that why not actually use this matrix, the same matrix, but put little spheres around each of the individual known binding site, and then by this kind of shape, find those regions in the which can be predicted as potential binding sites for this transcription factor. So in fact, why it's P? Because there is another algorithm which is called patch, and we'll look at it as well, which is very simple. They're saying, forget about positional weight matrix, just the true things, the true facts are the true sites. And that's what to do is simply, if you have your sequence, simply match true sites, the whole sequence of known site, and if it matches, then the new predicted sites should be also functional. But of course, this patch algorithm is then very conservative. So basically, we will find only those sites if we not allow any mismatches, which are exactly matching known sites. And we obviously will miss some unknown sites which are similar to known sites, but not yet found in this way. So that's why pmatch is actually is a kind of extension of this patch toward match. So it's somewhat in between. And our experiments were here demonstrated, in fact, that for about half of the content of the TRANSFAC, so if we consider all position weight matrices in TRANSFAC and compare using match and pmatch, only about for half, actually pmatch gives a priority and it gives a better false positive error rate compared to the match. So for instance, in this area, this is false negative rate, which means, for instance, if we here, it's 0.1, which means false negative rate is about 10%. So which means we will lose 10% of known sites, and we'll find 90% of them. With such a cutoff, the p-match gives an error rate of false positives around 0.2, but the match gives like 3 times higher false positive rate. Although for another area of false negative, the p-match can actually behave worse. So that's it's just another algorithm, and that's makes sense to try both and use it. So which we are going to do today. So I actually have to find my sequence again. We can start this p-match, and we will put. So we take it again, and we go here. We put it here. All right, and vertebrates, we take minimize. Actually, that's if we select now minimize false negatives, so which means when we run it, is remember with much, we found like more than 1,000 sites. But here, since we have additional pressure of selecting to be similar to true known sites, this will actually remove a lot of false positives. So that's why let's try this false negative, and we submit the form. Still, we found quite a lot of sites, but it's only 265. So it's a 5 times smaller number of sites than we found with simple match. Basically in the graphic, you see still a lot of coverage, but it's much smaller than if you just run the match. So why I still want to find more sites? Because if we miss some true sites, we of course can be wrong in interpreting how the genes are in fact regulating. All right, and then the last algorithm which I want to show today is called AliBaba, and it was also published some years ago. We will see it. There is a paper published in Silica Biology, and the idea of AliBaba, it goes even further after p-match. It actually asking a question: why do we consider each time the same matrix? So you see all these sites coming from the same transcription factor, but the matrix, which is actually gives kind of rules or regulations how the new binding sites could be found, are always considered the same. So why not actually change the positional weight matrix dynamically depending which place of the sequence you're looking at, and build those matrices on the fly? And that's what AliBaba is doing. So we'll try now this program. It's we go further to AliBaba 2.0, and we put here the sequence, and actually all right, so if we in the sequence, if we now start, so it's actually presenting here how it is doing it. If you see, it has those predicted binding sites for transcription factors, and now we are talking about the transcription factor name. So it's not a name of a particular matrix or a particular motif, it's a name of a transcription factor. And how it is working, it's in fact taking from a TRANSFAC library the true binding sites, and it tries to align them to a given sequence. And as soon as it aligned enough sequences from that side, from TRANSFAC, which should belong to the same factor family, then it tries to build a positional weight matrix from those sequences on the fly, and then estimate the score of that predicted binding site. And you see, although it finds many sites, but the overlap is much smaller if you compare it simply to the match. So, and actually, you can get this also as a list, which you can now, output as a and use it for your further purposes. And what kind of purposes you are going to use? And that's something we are going to discuss today. But before going to what is not done by by generegulation.com site. Let's consider the last thing, which is very important, which is an understanding of the site combination. So let's have a look at this picture. Basically, if you take a promoter region, and here on this picture, in all three lines are actually the same promoter. It's a human TNF alpha promoter in all these 5 cases. But it's an experimental fact from discovered from different papers that in different cell types, in T cells, in dendritic cells, in mast cells, actually the same place of that promoter, so the region from -107 to -74, is actually occupied by different transcription factors. For instance, here NFAT is sitting in mast cell, but in dendritic cell, the same place is occupied by an NF-kappa B. So which means all these overlapping predictions are not really some kind of artifact. They are really explaining and showing what can happen in reality. But then, which binding sites should we consider? And that would depend on which conditions we consider, which types of cell, which types of tissue. But even before going to that, very important concept is that transcription factors are not working alone. They are working in so-called composite elements in compositions, like in this case, the NFAT transcription factor work together with another factor, which is called AP1, and the binding sites for these 2 factors are close to each other. And in fact, this can be extended even further. We are talking about composite modules, so more, not just pairs, but more bigger combination of sites should be found in the regulatory regions, so that transcription factors which are binding to those places, they have to cooperate. So they bind to the nearby by sitting sites, and they synergistically activating the promoter by interacting to each other. So, and using this algorithm, we can find out of these thousands of binding site predictions, we can find actually those combinations which are really responsible for a particular function. So, and there is an algorithm which is published in 2006, but that was actually developed in many years, which applies a genetic algorithm and tries to find those combinations of binding sites. And this algorithm is called CMA, Composite Module Analyst, and it's also available in our generegulation.com. And here it's called Composite Module Analyst. And here it shows what it's actually doing. It's searching for this combination of binding sites using genetic algorithm. And in fact, what we are going to do here, we are going to apply it to a set of sequences which are commonly regulated. So let me actually take this from my files. All right. So I took promoters of 22 genes which are activated, upregulated upon immune response. Actually, there is an experiment. A cell line was treated by a molecule called TNF alpha, which triggers a very important response. Several genes are highly upregulated, and it's known actually how the mechanism of that regulation. It's done through the transcription factor called NF-kappa B. But taking the data from that experiment, we can extract promoters of 22 genes which are upregulated upon TNF alpha stimulation. So, and then this algorithm, Composite Module Analyst, can analyze not just one promoter, but it can analyze all these 22 promoters together. So we can put it here. But it needs a comparison. It needs to compare to some background set. And as a background set, we choose actually a set of random sequences. Here, well, not completely random, not binomially generated random sequences, but actually I generated random sequences from just randomly picking up them from different promoters in the whole genome. So let me see where I have it. Yeah, here. So here there are 50 randomly generated random sequences in human genome. So we can copy them and put them here. And then there are a couple of other parameters we have to set, which are important for us. Actually, it's looking for how many binding sites we should find in this composite model. How many binding sites for transcription factors should be together in a one window, so that the transcription factors which are interacting with them, they basically regulate the gene. So, and here we have to set these composite module parameters. We, for instance, can say the number of sites should vary from 3 to 6, and the number of pairs, it's look also for very close pairs, which are sitting very close together. Let's search also from 3, let's say, to 6. And then there's, this parameter is called the profile, actually collection of matrices we are going to use. And we will select vertebrate non-redundant profile. This is a special collection of matrices which cover most of the transcription factor families. And then we have to generate iterations, and this algorithm works in iterations, and the number of random generated models, it's called population, let's say 200 and 200. And then we run it. Well, it takes a bit time. Actually, I will show you the results of that run. And the results is here. You see it found these 3 motifs out of so many. You remember how many motifs and binding sites were found only in one promoter, but now we have 22 promoters. But it selected this OCT1 and NF-kappa B, 2 NF-kappa motifs, and also these 2 pairs of CREL, well, it's another NF-kappa B family member motif and glucocorticoid receptor, and YELK1 and LIF1. And here is this distribution shows you how this this composition of these motifs is found frequently in the promoters of our 22 genes. These are red boxes, compared to the randomly picked up genomic promoters. And you see these 2 distribution are clearly discriminated. And on this window, you see basically each promoter of each gene. And this is, by the way, our gene, CXCL1. And in this promoter, it found this only 2 sites for NF-kappa B in this case. But there are other promoters like this one, which have actually higher score. It has it found this composite module in this place of promoter. So what it means that actually, although the promoter, for instance, for this gene has a lot of other sites, so potentially a lot of other sites are present in that promoter, but this part, this composite module is acting to regulate this gene upon TNF-alpha stimulation in particular condition. And that's the advantage of that composite module analyst. So it can actually reveal those transcription factors which are working in particular conditions. All right. So, but that's, so what? Actually, the question, so what, is always is important for us. So we found this way or another way, we found transcription factors which are important for regulating my genes. But now, so what? And basically, a very important point is to ask a question how these transcription factors are actually regulated. So we ask this question. So we ask a question, okay, these transcription factors are regulating my genes, but who is regulating activity of those transcription factors? We call them master regulators. And this master regulator search is not easily, is not possible to do on this generegulation.com. So it's the generegulation.com is a very nice site for your first steps to find those transcription factors which are regulating your genes. But now to ask a question, what are the master regulators? Why do you need to ask that? Because once you find those master regulators, you may use them as a drug targets, as biomarkers, as some central points of the mechanism, how your genes are actually regulated. What is the most important mechanism of regulating my genes? And that can be done using another free tool, actually, which is called the GeneXplain platform. And I will show you just a little glimpse of it. So if you go to our website, to GeneXplain website, geneXplain.com, not generegulation.com, but geneXplain.com. And here you see this free platform. You can click here and to do the free registration. And once you done this registration, you get such a you get your password and the username, which is different from the generegulation.com, so it's a different service. And then by clicking on this GeneXplain platform, you will get basically something like that. And that's another the whole talk how to use this GeneXplain platform. But what I want to show you that using this GeneXplain platform, you can take the results of generegulation.com, you can take the results of predicted transcription factor binding sites, and try to find master regulators which are regulating my sites. So just briefly, basically what I did, I copied all predicted binding sites from the match, so that they are here. So in this table, let me open it. So these are the same sites which you've seen on that generegulation.com. I simply cut and paste them in Excel table and imported them here in the platform. And then I converted each of that matrix in the transcription factor. And that can be done also, step by step, and you do it in the generegulation.com, because each of the matrices is linked actually to transcription factor. You cannot do it in that way by one batch. So you have to do it, step by step. But eventually you get these transcription factors which are linked to these motifs in a different table. So these are my transcription factors, 100 transcription factors, which are linked to my table, and some of them, like this case, they're linked to the same matrix. And then using tools from that platform, basically, if you open this platform, you see this very colorful interface. And if you go here to gene or protein list, so now you want to find master regulator from my transcription factor list, my 100 transcription factors. And there are two ways of doing it: using TransFAP database, which is a licensed, so you have to license it to get access to it. But there is a free database called GeneWays, which is based on text mining. So the TransPass is based on manual curated collecting interaction of signal transduction network, but GeneWays is actually built using the automatic algorithms. And then you click on this pipeline. So you see here a pipeline, and you take this gene list. Well, I converted it in fact into n threads, and you run this workflow. And as a result, you find this. Let me open it broader. This network. You see these blue guys here are the transcription factor, the 100 transcription factors, which are found by the match algorithm using generegulation.com. And this pink one are the master regulators, which are expressed in that, which are upregulated in that experiment, and they are able to control activity of those transcription factors through this network. And network, again, is taken from the database called GeneWays. So this master regulator network can be modified. You can put a different layout on it, and you can export it in different formats. And basically, just try your platform account, and you will be happy to do actually next step. To not just find your binding sites, but really to find master regulators. And all this is free. You just can do it from what is available and what is provided. Of course, just in the last 5 minutes, I promised to finish in 40 minutes, but now it's already 50. I'm sorry, we just the last 2 minutes. I want to show you actually what you get when you have the full access to TRANSFAC. And that you get actually, by licensing it. In fact, then you can apply not simply these small pipelines, but you can actually apply a bigger pipeline doing really an extensive multi-omics analysis. So we have here, we have a very complex tool called Genome Enhancer, and it can analyze multi-omics data in one click. So you don't need to cut and paste and combine different tools and run them. You simply upload your different types of omics data, and in fact, you see there is a number of projects here. And for instance, here there is a data coming from gene expression study on rheumatoid arthritis, where actually a lot of immune response genes are involved. And at the end, this tool is just by clicking through, is generated a full report, which involves not only prediction of binding sites, it actually uses a much bigger library of predicting binding sites for transcription factors. You see it's actually building very similar modules like the CMA is doing, but with a much more broader library of motifs. And then it builds actually a network with master regulators like this, using a manually curated database called TransPass. And this TransPass is actually taking not simple interactions like gene activation. It's taking into account complex mechanisms like phosphorylation, complex formation. So all these complex steps which are happening in single transduction are analyzed here, and then you get these master regulators. And at the end, it even goes to predicting potential drug targets and even drugs which can be applied against those master regulators in the context of particular disease. So just to give you a little glimpse of what you get when you go even next step, and you apply a licensed TRANSFAC. Good. Okay. So I think with that, I will finish my presentation today, and I see a number of questions. First question is, I suppose I want to find the promoter enhancer sites for HBB gene. First, is it known? And if not, how can I predict it? All right. So, all right. And there is another comment. How do we complete the promoter sequences of differentially expressed genes? Also, what about the genes that are repressed, reduced expression? Very good questions. Thank you very much. So actually, concerning the second question, repressed genes, yes, we of course analyze it all together. So for instance, in this example, you see we took gene expression data, and the algorithm reveals upregulated genes and downregulated genes separately. And then it analyzes promoters of those two sets of genes separately. It builds a composite module for upregulated genes. So, and you see that, for instance, it makes sense. We know that upregulation in immune response is based on activation of NF-kappa B, and you see these two or even three motifs for NF-kappa B found in this module. Also, you remember there was a glucocorticoid receptor, interferon response factor. So that's all make a lot of sense for genes, upregulating genes in immune response, but it's actually also taking the downregulate promoters of downregulated genes and builds another module for regulate, which discovers a module to regulate these downregulated genes. So that's done by this Genome Enhancer, but the same can be done, of course, but more like step by step, manually, using these tools here in this generegulation.com. So by Composite Module Analyst, you actually have to put here the FASTA files for the upregulated genes versus random background, or promoters of downregulated genes versus background. And as a background, actually, you can, in fact, use random background, but if you have a differentially expressed genes and you have data of differentially expressed of RNA-seq or Affymetrix array, so you, in fact, though, if you consider all those data, you have also genes which didn't change expression. So they are in the same condition, but they didn't change expression. So that would be the best, background set, the promoters of genes which are from non-change conditions. Now the first question was how to extract those promoters, and that's basically even it can be done also here in the free platform. For instance, here we have our set of genes. So this is a set of our genes, right? And we need promoters of those genes. Let me open them. So these are 22 genes, and here we have symbol IDs of those genes. Some other columns which I just get from a database. These are gene symbols. So and in this tool, in this free platform account, you can actually extract promoters of those genes. You can extract promoters of all genes or just for one gene. So here we have this tool called, let's see, it's, I think, inside analysis. No, it's here. It's called gene set. Select gene set to track. You see how many tools we have here. Sometimes it's not easy to find them. Yeah, super. Track, not track to gene set to track. Yes, this one. So basically, you take your gene set like this, you drag and drop it here, and then you specify these promoter areas, for instance, 1 kb upstream, 100 nucleotide downstream, and then you run it, and it goes to the genome, to the latest human genome, and extracts promoters of those genes here, and then plot them on genome browser. By the way, you can actually put here also the genes, respective genes, so different tracks like gene track can be put here. So we have here this promoter of that gene, and there is another promoter somewhere. Yes, there is another promoter of that gene. So you see this location of those promoters. By the way, if you have your sites here, you can also put those sites here. No, they were not predicted in that promoter, but then you can see that's not in this chromosome. I think there I was predicting them in chromosome 4, right? So then you see those sites which were predicted actually by generegulation.com, by the match algorithm. Here you can put them in the context of the genomic context, actually going down to the nucleotide level and see the each site and the score of that site and so on. So, but the question was, I always like deviating, all right? So the question was how to get those promoters. So now we have those promoters as a track, and we can export it here. Let me close it. With a right click on the file, you can click export, and you can export it as FASTA sequences in your computer. and then bingo, you have your FASTA sequences of the promoters of your interest. All right. Okay, there was another question. Could you please suggest a reliable database or website where I can check whether a DNA binding protein from bacteriophage has known binding specificity? I'm trying to find out if it's in transcription factor or has a defined DNA motif it recognizes. Can your site be used to give you some info? All right, so it's from bacteriophage, of course. Our databases are focused on different eukaryotic organisms, mainly human, mouse, and rat. But also some other organisms, some other species, but no prokaryotes, even bacteriophages. So basically you won't find in TRANSFAC any information about proteins from bacteriophages. But the one thing you might be interested to look at is transcription factor classification on our website, which goes which is built by Professor Wingander, who is actually a very important figure in the transcription factor, theories and practice. So where was it? Yeah, it's among free tools, and here we have classification of human transcription factors, and here you have all the proteins and their uniprot IDs here in this big table. It's a quite comprehensive list of human transcription factors. But of course, in your case, you're interested in bacteriophages, so you won't find it here. Sorry about that. All right. Another question. Can I predict nucleotides positioning as well as TF with CMA? All right, so I'm not sure if I understood the question correctly. So nucleotide positioning. Maybe you mean the revealing the motifs de novo. That's also possible. So de novo. So basically, in what I was talking so far was about TRANSFAC, and where we collect known motifs, so motifs which are already discovered, and our algorithms, all the match and CMA, are actually operating with those known motifs. And as when talking about human, or we can say that we made this estimate some time ago, about 93–95% of all human transcription factors have at least one motif in TRANSFAC database. So which means there are very few unknown motifs for human transcription factors. But this, of course, might be different if you are studying some other type of organisms where the transcription factors are not well studied. So in such cases, it may have a, it may be interesting to apply the de novo motif discovery algorithms, and indeed we have some of them among our tools, and they are available for free also in the free platform. So among the methods in the site analysis, I think I think it's in NGS folder. We have these actually tools called ChIPHorde and ChIPmunk, which are very nice tools to do this the de novo motif discovery. So basically the same thing. You take your promoters and you put them here, and then this algorithm will try to build the motif de novo or several motifs de novo. So there is another tool which I think I think the ChIPmunk is for one motif and ChIPHorde is for several motifs. And those motifs are actually discovered and saved here in the platform as positional weight matrices. And you can actually mix them together with TRANSFAC matrices and run them the search for new sites using this matrix, which is just generated by you, together with known motifs from TRANSFAC. Sorry, nucleosome positioning. Sorry, it's completely different topic, but hopefully what I was talking about was making also sense for someone here. So no nucleosome positioning sites we don't have. That's a big debate whether nucleosomes actually have any preference in binding motifs to DNA. And some people reporting the specific motifs for nucleosomes. If they exist, they are very vague. So by definition, we are not considering nucleosomes as transcription factors because there is a very particular definition of transcription factors, which actually Edgar Ingender published in this book many years ago. Actually it tells that it should be a protein which binds specifically to specific places in genome, but it should be involved in specific regulation. So like, for instance, regulating genes in specific conditions. To that, in that case, it's hard to say that nucleosomes can be classified as transcription factors. All right, so I think, I'm still open for your questions. I don't see at the moment any further questions. All right, so I hope my short introduction and very quick talk was useful for you. So in case if you are still interested in this topic, so we have this, we are going to have this seminar recorded, and it will be put in our Academy website. So where this GeneXplain Academy, we call it No Coding Bioinformatics Academy, because we try to use developed user interfaces that you don't need really to code. You just drag and drop your sequences, you change your parameters, you run, you get results. So so that's here. There's an announcement of that seminar today, and you will find here also the recording of that seminar together with early seminars we had the last year, and we have quite a lot of seminars devoted to various topics, and in most of them we used actually I think in all of them here last year I was talking about how to use our commercial tools like the full TRANSFAC to do many things, like to analyze the DNA methylation data, ataxic, cut and run, multi-omics data, analyze mutations, many different things. Just look here, you find description of those seminars and also the recording in YouTube, which you can watch and understand how our tools works, and maybe it will be useful for you. Have you seen any fruitful efforts of using AI in this area? All right, very interesting question. And in fact, I'm going to have a seminar about AI. Let me say that indeed, it's now a very, very interesting time. So far, those attempts of applying AI are still quite vague, but we can see it's developing very quickly. And especially, I like those approaches which are around these, larger language models. And there are a few algorithms already now available, which are applying these large language models similar to ChatGPT, but applied to a DNA language. And they have already quite a good progress in predicting interesting things. So still, in a direct competitive comparison. So such a knowledge-based tools like TRANSFAC, we collect knowledge for many, many years, is still winning because it's it's based on a very solid knowledge. But who knows? Maybe in future such tools can overrun. Although I think our future is actually in merging these two approaches together, applying AI algorithms and not throwing away the collected knowledge so far, but really merging these two approaches together. And hopefully I will talk about that in near future. Just watch our announcements. All right, I think I don't see any further questions. Thanks a lot for your interesting questions, and see you next time. And have a nice, day. Goodbye.